Hello, my name is Jesse White. This is my critical assignment preparation 1 assignment, and this is ATP pathways for CAN 5.6. We'll go ahead and jump right into it. All right, so up first, we're going to talk about glucose metabolism. So the two main things to focus on here is before glucose, which is that primary singular source of sugar as energy, can be put through the citric acid cycle or the Krebs cycle, has to go through the two processes of being metabolized down to pyruvate and then once again to acetyl-CoA. And throughout this whole process, as it's being metabolized, there are coenzymes NAD and FAD down at the bottom once we get into the Krebs cycle that are removing those hydrogens that are coming off during the metabolization of those molecules, and it's being pushed over to the electron transport chain. So once again, glucose is moving down to the 2-pyruvate. If there is no oxygen present, and this is an anabolic process, then that pyruvate cannot move on to the acetyl-CoA, and that will eventually end up turning it into lactate. So as it says there, as it points out, where does that lactate go? It'll go back into the blood, back to the liver, and it can actually be processed back in to be used as energy later when oxygen is present to metabolize to acetyl-CoA. Once that's broken down to acetyl-CoA, it has that permission to enter the Krebs cycle, and it will do this for 2 full rotations, again dropping off those hydrogens as it moves through the system, through the NAD and the FAD coenzymes, dumping those off in the electron chain, transport chain. And what this does is it keeps the acidity of the whole cycle and the chemical reaction process safe. And also you're seeing a drop of 4 CO2s and a total of 2 ATP produced in the Krebs cycle portion, and then a net of 2 ATP during that initial phase. That'll bring us to a total of 32 ATP produced from the glucose metabolism. So fatty acid metabolism, it's kind of has the same principle. It's a little more complex. There's some extra steps involved, but once again, following that general guideline of having to have that acetyl-CoA as the key into the door to the Krebs cycle, we have to break down to that point. And the way that happens is through lipolysis, those triglycerides are broken down into their most single basic form, which is 1 glycerol with 3 free fatty acids. Once the glycerol is broken down, it moves over to the glucose, and it's pretty much going through that same process of the glucose metabolism that we talked about earlier, while the 3 fatty acids will go through something called beta oxidation, which is that process of taking those fatty acids, breaking them down, metabolizing them further to produce those acetyl-CoA 2 groups. So depending on the carbon content of the fatty acids, so with palmitate we have 16 total carbon fatty acid for that one. So they'll be breaking off into 2 by 2 chunks slowly as they get removed. So that'd be 8 2 acetyl-CoA groups pushing over. And then they would rotate through the Krebs cycle once again. And again, these free fatty acid pools are coming from those fat stores within the body, and that's Kinney et al. 2020. So again, the yield for this is going to vary per that carbon content that you find with whatever those free fatty acids are. And just the instance or the example that was provided was that palmitate has that 16 carbon fatty acid that would yield a ATP yield of 106. So some pathway limitations and considerations, and we're talking about these two. Just looking at this diagram right here, this was a good diagram I pulled from a book I had from another class. It's a Kinney et al. 2020. Excuse me. There's fats, carbohydrates, and proteins. There's 3 methods that we can pull from that food intake. So not one source of metabolism is going to take place. It's not going to take full reign over everything. There's going to be kind of a pull from all three of these throughout whatever the training event is. So some of the limitations of glucose is that present of O2. So where there's going to be anabolic or aerobic metabolization taking place. So again, if there's no oxygen available, that pyruvate is going to be pushed into lactate and lactic acid. Whereas if the oxygen is available, it can go through the process and get kicked into the Krebs cycle. Another downside of the glucose is it has a very fast production time, especially with the anaerobic side, but it produces a very low yield. So it's easy to produce, but it does not produce as much. One of the limitations with fat metabolism is the oxidative capacity of the individual. So whatever their body type is able to do, so the fast twitch versus slow twitch muscle fibers, and their ability to go through the oxidative portions to create that acetyl-CoA to push into the Krebs cycle. So just the efficiency of their body in that process can be a limitation as to how effectively their body can produce or pull from that source of energy. That's Kinney et al. 2020. And again, this is a slower production, so it doesn't happen as fast, but it produces a much higher yield in the end result. Some other considerations to think about when we're talking about these different energy types. So the energy is going to be pulled from whatever the demand and duration is. So if it's maximum full effort right up front, like you're trying to pick a car up, that's going to be more of the anaerobic glucose side. And if you're looking for more of like the long distance endurance type exercise, it's going to be more towards the fat. That's where the energy is going to be pulled from. So they would take the proportion of those. And again, like I said before, the body type and somatotypes, Kinney et al. 2020, slow versus fast twitch muscle fibers and the body's overall effectiveness at the oxidative capacity. And another thing, just with this in general, back to the balance of the Krebs cycle with the hydrogen, is the significance and implications of hydration levels. So once that osmolarity is thrown off in the system, in the cells, it makes everything, all the gears kind of grind to a halt. There's a lot of complications. It can lead to imbalances with other electrons and everything else. So just keeping that adequate level of hydration is going to help individuals maintain that fluid motion throughout all energy metabolism pathways. I only used two references for this video presentation. That's Kinney et al., Physiology of Sport and Exercise, and then McArdle et al., Exercise Physiology, which is the book for this class. And that is all I have. And that wraps it up. Thanks.